Everybody, you're welcome to today's class, and I will be talking on the cell and its environment. Let's start. The first thing to note about cell and the environment is that we must first understand the concept of diffusion. And diffusion is the movement of molecule or ions from a region of high concentration to a region of lower concentration until they are evenly distributed. And the substance involving diffusion may be liquid, gases, or solid. So there's something you should know about diffusion. We are moving from higher concentration to lower concentration until they are evenly distributed. Just imagine, maybe you spray an insecticide into a room now, or maybe you spray a perfume. What will happen is that those gases that are inside the perfume, or the smell and the flavor of the perfume, will move from one place to another. We move from a higher concentration to a lower concentration. So just know that diffusion is the movement of substances from region of high concentration to a region of lower concentration until they are evenly distributed. And diffusion can either be solid, liquid, or gases. So now let's move to the factors affecting diffusion. The first factor that I have here is the presence of concentration gradient. And what this actually means is that there has to be a high concentration and there has to be a low concentration. There has to be presence of a high concentration and low concentration for diffusion to occur, because diffusion is the movement of molecule from higher concentration to lower concentration. Then we also have the surface area. Surface area also affects the diffusion. We have the state of diffusing substances. If you remember that we have three states of matter, we have the solid, we have the liquid, and the gases. And normally the gases will diffuse faster than those that are liquid. Then we also have the size of molecule or particles. So what this means is that substances that are smaller in size, will diffuse faster than larger one. Then we also have temperature. So something that we should know about temperature, that increase in temperature will increase the rate of diffusion. Increase in temperature will increase the rate of diffusion. Now let's move and look at the importance of diffusion to animals. That there is intake of oxygen or nutrients from the mother to the fetus through the placenta. Then we should know that gaseous exchange in mammals occurs in the lung during respiration, and there's movement of carbon dioxide from the lung capillaries into the air sac. All these three importance that I are seeing various more. I just list this one so that the video will not be too long. All these factors, all these things that are happening right now, they are because of diffusion. So we also have the importance of diffusion to flowering plants. The first one is the movement of carbon dioxide through the stomata into the leaf during photosynthesis. So water vapor leaving the leaf during transpiration. When we talk about transpiration, that is the loss of water in form of water vapor, and the movement of oxygen into the leaf through the stomata during respiration. All these are also done by diffusion. So now let's move to the second thing that must know, that is osmosis. Osmosis is defined as the flow of water from a region of lower concentration or weaker concentration or dilute concentration to a region of higher concentration or stronger concentration or concentration solution through a selectively permeable membrane. So there's something you should know about osmosis. You know that in diffusion we are moving from high concentration to low concentration, but in osmosis what we are actually doing is that we are moving from lower to higher, and this must be done through a selectively permeable membrane. There must be a selectively permeable membrane for osmosis to take place. So conditions necessary for osmosis to take place. Number one is the presence of a stronger solution. There must be a presence of a stronger solution. There must be presence of a weaker solution, and there must be presence of a selectively permeable membrane. So these are the three conditions for osmosis to take place. And then we should know something about in osmosis. A lot of time for osmosis to occur, there are usually two solutions. Sometimes there are usually two solutions, and these solutions are separated by a selectively permeable membrane. The weaker solution is said to be an hypotonic solution. So the moment you hear hypertonic, it means that the cell is surrounded by a low concentration. While the stronger solution is said to be hypertonic. Whenever you have a cell and that cell is surrounded by a stronger concentration, it actually means that the cell is hypertonic. And then when the solution have the same concentration, it means that they are isotonic. From the word iso, the meaning of iso means the same. It means that they have the same concentration, and because they have the same concentration, there will not be net movement of water. For example, now when a cell is placed in an hypotonic solution, and water from the outside environment will move inside the cell. But when the cell is placed in an hypertonic solution, water will leave the cell out to the outside environment. But when the cell is said to be in an isotonic solution, there is no net movement of water. So now let's look at the differences between diffusion and osmosis. The first difference is that diffusion occurs in liquid, gases, and even solid, but osmosis only occurs in a liquid medium. And also diffusion does not require a semi-permeable membrane, but there must be a semi-permeable membrane for osmosis to take place. And also diffusion occurs in living and non-living things, but osmosis naturally occurs in living things. And then the fourth difference, that both the molecules of solute and solvent can diffuse, but under the osmosis only the solvent molecules can diffuse. So these are the differences. You can just master this, although there are still more, but for today, for the sake of this class, just master this class. These are the type of question that you should know when you are preparing for your WAEC, NECO, for your external exam, even your post-UTM. These are some basics, and sometimes it is funny sometimes that students are funny sometimes that they forget these basics. That's the reason for this class. So now let's look at another thing to know about the cell and its environment, about a condition called plasmolysis. When we talk about plasmolysis is defined as the outward movement or flow of water from living cells when they are placed in a hypertonic solution. You know that I said it before, that when a cell is placed in a hypertonic solution, that means the environment around the cell is a stronger or higher concentration. I know something that water will move from lower concentration to higher concentration. What that means is that water will leave the cell to go to the outside environment. And when it goes outside to the outside environment like that, that phenomenon is called plasmolysis. So the plasmolysis, in the process of plasmolysis, involves the withdrawal of water from living cells to the extent that it will result in the pulling away of the cytoplasm from the cell membrane or the cell wall. It will pull away the cytoplasm, and when that has occurred, we said that cell is plasmolysed. So now let's look at another thing that we should know about cell and its environment, and that is hemolysis. When we talk about hemolysis, this is defined as a process by which red blood cell burst as a result of being too much water passing into it. So this is the bursting of red blood cell when there is much water passing into it, and this occurs when the cell is placed in a weaker or hypertonic solution. When a cell is placed in a hypertonic solution, water will move from the outside environment, because that outside environment like that now has a low concentration while the concentration of the cell is high. So water will move from a region of lower concentration to a region of higher concentration. So water will now be moving inside the cell. Water will now be moving inside the red blood cell, that will now result in the bursting. So that bursting or splitting is called hemolysis. So now let us now look at turgidity. The turgidity usually occurs in plant cells. So turgidity is a condition in which cells absorb plenty of water to a point where the cell is fully stretched, and this occurs when the cell is placed in an hypertonic solution. So one major a lot of time why bursting does not happen in plant cell the way it happens in animal cell is that plant cell has a cellulose cell wall, and the cellulose cell wall that plant has is very strong, so the cell will not burst. What will just happen is that the cell will become rigid, and that rigidity is called turgidity. So the condition whereby cells absorb plenty of water to the point where they are fully stretched, and it occurs when they are placed in an hypotonic solution. So turgidity is useful to the plant because it makes them stand erect, give support to the stem, leaves, flower, and guard cells. So the guard cells are actually the cells that are responsible for the opening and closing of the stomata. Now let's now move to the last thing that I want us to know for today about the cell and the environment, that is flaccidity. From the word flaccid, the moment you hear the word flaccid, it means that this thing has lose water. So flaccidity is defined as a condition in which plants lose water to their surroundings faster than they can absorb. They lose water to their surrounding faster than they can absorb. That is the meaning of flaccidity, losing water faster than they can absorb. And flaccidity usually occurs when there is no water in the soil or during droughts, and sometimes it causes the wilting of the plants. So this is the end of today's class, and I really appreciate us for listening so far. And please don't forget to like and subscribe. Thank you very much. Have a wonderful day.